Howdy, my name is Jazlyn Gonzalez and I am part of group number 6 with Andres Garza. Today we will be presenting over Lab 2 aseptic techniques. So a quick list of our contents is the introduction, materials, procedure, our observations and results, and our conclusion. Aseptic techniques are crucial in microbiology as they prevent contamination and ensure the accuracy of our results. Without proper sterile technique, unwanted microbes can interfere with cultures, which leads to inaccurate results. Proper aseptic techniques protect us from exposure to potential pathogen organisms. With these techniques, we can reliably isolate, identify, and study microorganisms. Now for our materials, we use 2 tryptic soy broths, also known as TSB. We use 2 tryptic soy agar or TSA in 3 milliliters. We use 2 tryptic soy agar or TSA in 9 milliliters. We use 3 tryptic soy agar TSA plates. We used a loop inoculator. We used a needle inoculator, a Bunsen burner, Escherichia coli or E. coli in TSB, Staphylococcus aureus, also known as S. aureus, on blood agar plate, and mixed culture broth. Step 1. Inoculate a TSB using a loop with E. coli. Step 2. Inoculate a TSB using a loop with S. aureus. Step 3. Inoculate a TSA 3 milliliter using a loop with E. coli. On small slants, just streak the slants. Step 4. Inoculate a TSA 3 milliliter using a loop with S. aureus. On small slants, just streak the slant. Step 5. Inoculate a TSA 9 milliliter using a needle with E. coli. Stab into the butt of the tube and streak the slant as you pull the needle up. Step 6. Inoculate a TSA 9 milliliter using a needle with S. aureus. Stab into the butt of the tube and streak the slant as you pull the needle up. Step 7. Inoculate a TSA plate using a loop with S. aureus. Streak for isolation. When coming from a solid media, just use one colony. If the colonies are extra large, just touch the colony. Step 8. Inoculate a TSA plate using a loop with E. coli. Streak for isolation. Lastly, step 9. Inoculate a TSA plate using a loop with mixed culture broth. Streak for isolation. From this media on Tuesday, you should see 2 different colony types.
Moving on to our results, we're going to be looking at S. aureus on the TSA plate. So for this one, we have larger colonies with convex dome-shaped elevation and creamy opaque texture. More so, their golden yellow pigmentation was also caused by the staphylococcus which distinguished them from smaller, smoother E. coli colonies. As for the E. coli on the TSA plate, colonies were small to medium-sized and also circular with smooth edges. They also had a moist, glistening texture. So this appeared grayish to white to an off-white and were slightly translucent, which is typical of E. coli. And lastly, as for the mixed culture on TSA, the streaks from the inoculation overlapped, causing some of the colonies to blend together. And while E. coli and S. aureus were present, the overlap made it a bit difficult to isolate and clearly distinguish between the 2 types of colonies. Moving on to our other results, we can see that for E. coli and TSA 9 milliliter, there is a growth that's visible along the stab line that's spreading up with the slant surface. So since E. coli is a facultative anaerobic, it will grow on both the stab line as well as the surface. As for S. aureus and the TSA 9 ml, we can see that there is dense growth along the stab line and across the slant. S. aureus is also facultative anaerobic, so growth occurs throughout, but often heavier at the surface. And lastly, as for E. coli on the TSA 3ml, we can see that there is moderate bacterial growth with a smooth, thin, and cream-colored growth spreading across the slant. As for our results on S. aureus and TSA 3ml, it shows that there is moderate growth along the streak line as well as a slight spreading into the medium. As for E. coli and TSB, we can see that the broth appears uniformly and it also appears cloudy since E. coli disperses evenly throughout the liquid. And lastly, for S. aureus and TSB, both appeared turbid with flocculent clumps that settled at the bottom. The reason for this is because S. aureus tends to cluster. So, in conclusion, this lab emphasized the critical role of aseptic technique in microbiology, as it allowed us to transfer microorganisms with different media while minimizing the risk of contamination. Through inoculating broth, slants, deeps, and plates, we observed clear differences in growth patterns and colony morphologies of Escherichia coli, otherwise known as E. coli, and Staphylococcus aureus, S. aureus, which reinforced the importance of using proper tools and streaking methods. Although overlapping streaks on the mixed culture plate made isolation more challenging, the results highlighted the need for precision and consistency in technique to ensure accurate identification and study of microbial species. This page is also just to provide the references that we used for completing this lab. This concludes our video presentation, and I want to thank you guys for watching. And I also hope this helped you better understand the importance of aseptic inoculation techniques in microbiology.